Just to go a little bit deeper into DNA, it stands for deoxyribonucleic acid, and it's in the form of a double helix, which we'll see later. It's located in the nucleus of the cell, and the chemical bases are attached to a sugar molecule and a phosphate molecule, and the base, the sugar, and the phosphate molecule together are called a nucleotide. The four chemical bases are adenine, which is represented by letter A, guanine by G, cytosine C, and thymine T. And then those pair up with each other, and A pairs with T, and C pairs with G, and those are called base pairs. Long strands of those together form the nucleotides, and those then form the double helix. So here's just a few pictures. So I have a picture of adenine and thymine bonding together, which takes 2 hydrogen bonds, as you can see, and then guanine and cytosine bonding together, which takes 3 hydrogen bonds. And then we have this picture, which just shows A and T, C and G binding together and forming that twisting double helix. Okay, so we're going to do a little example of matching the bases. So I did remind you that A binds with T and C binds with G. So Allie, will you please come to the board, and I want you to write the original strand, and then I want you to write the matching strand underneath it. Through it really quick. So the original strand was A, G, T, C, C, A, T, T, G, A. And so we said A pairs with T and C pairs with G. So she did A with T, G with C, T, A, C, G, C, G, A, T, T, A, T, A, G, C, A, and T. So she matched them all correctly. So good job, Allie. Okay, just to sum up kind of DNA, genes, chromosomes, those all kind of get jumbled around, mixed together. You know what DNA is, so we talked about our strands up here. The specific segments of DNA coded with instructions for a trait are called genes, and then we bundle those all together to form chromosomes. So Punnett squares, those are kind of scary, but we should be able to learn this pretty easy. You use the 2 sets of alleles from the parents, and you combine those to form the genotypes of the parents' offspring. So that would be what kind of makeup their children are going to have. And we're going to do a little example. Okay, so our 2 parents have little a, which would be homozygous recessive, and big A, which would be homozygous dominant. And so we split them up, and we have the pair on the right on the top, one big A, and another big A. And the pair on the left, we have down the side, so we have little a, little a. And then we're just going to drop down the letters from each square and combine them, putting the uppercase letter first, always. And so we have big A, little a here, big A, little a here, big A, little a here, and big A, little a here. And those are going to be all heterozygous because one letter is dominant and one letter